Thank you everyone for joining us this webinar with PPMD and Wave Life Sciences. I'm Eric Camino, I'm PPMD's Vice President of Research and Clinical Innovation. And today we are joined by Paul Bulna, the President and CEO of Wave Life Sciences. And Paul is going to walk us through some of the 48-week results from FORWARD-53, which is Wave's trial for WVE-N531, which is their investigational exon skipping product for patients amenable to exon 53 skipping. As we go through the webinar, if you have any questions, we will have an open Q&A at the end, so please submit the questions in that chat box that's at the bottom of your screen. We'll try to get through as many of those as we can. We did have a few submitted ahead of time as well, so we'll get to those ones with the open Q&A. We are recording this webinar as well, so if you have to step away or if you have family members who may benefit from watching this webinar, it should be up on PPMD's website within the next week, so feel free to share it with them, or you can come back and watch other pieces if it's helpful. But with that, we're going to get started for today, so I'm going to hand it over to Paul, and we'll hop in.
Thank you, Eric, and we appreciate the whole PPMD team setting this up so quickly following the data. I think it's an important way for us to connect with the community after these exciting data. Before we start and share data, I will say that we will be making forward-looking statements during this discussion, and it's important to remember this is still an investigational product. I think stepping back, we've had now an over decade-long experience working with you, the DMD community, boys, families, organizations, and it's really important that at the center of what we do is this engagement with the community. You should know that why we get up every day and do the work we're doing, what propels us forward, are the experiences we have with you sharing your stories with us. And I think it's incredibly important to realize the impact that that has on continuing to give us the energy we need to continue to make important medicines. So with that, we'll share a little bit again about the preclinical data that brought us into the N531 clinical program, and then we'll jump into the 48-week data. But it's really important as we step back and began the WVE-N531 program, we really did ground this in important animal model work in order to predict and design our clinical progress. So stepping back, we're a firm believer in the double knockout mouse. We use that as the mouse model to move forward with. Over the years, we have used other models like MDX, but recognizing that in the double knockout mouse, there's no utrophin knocked in, meaning we're only looking at a model system where dystrophin is depleted, and ultimately that impacts the mouse in terms of its survival characteristics. We felt that that was an important model to use in designing our preclinical path forward. Therefore, what was exciting about those data early on were they were giving us high levels of dystrophin, not just in skeletal muscle, but in heart and diaphragm. In those data sets early on, and we've shared these data on webinars in the past, one of the things that was impressive was we were able to see improvements in respiratory function in that animal model, meaning we could restore levels back to wild type levels and ultimately see 100% survival in this mouse model out at 40 weeks, which was the end of the study, whereas typically these mice die at about 8 weeks. So it's a very rapidly progressive model with which to study. But we felt a really important one because what distinguishes that survival characteristics is the ability to produce dystrophin, functional dystrophin, and ultimately see that translate. In addition to the work that we have done in the double knockout, we also did work in the non-human primate. And that was really looking at how do we get exposures and different differential concentrations, meaning, as you can see, higher levels actually of drug concentrations in heart and diaphragm than even in the skeletal muscles. So again, getting that exposure, and I should say all of this is really grounded in our oligonucleotide chemistry. And what's different about the ability that we have with our chemistry, and in particular what we call PN chemistry, is the ability to get this delivery and these exposures without utilizing conjugates. So we don't have to take on additional liabilities with various conjugate structures, but our oligonucleotide delivers to these tissues. So those were the data that helped support moving forward. We then moved forward into the clinical study and translated that. This was FORWARD-53. This was an open label study, and it had several components to it. And it was important for us as we came back with N531 that we looked in an exploratory way, in an experimental way, to see how does that data that we saw in the preclinical setting translate into the human clinical setting. And so part A was designed to be exactly that, an exploratory study. There was an ability to look within 3 boys at 10 milligrams per kilogram with 3 doses. So basically, it was a very short study. It was a 6-week study. And what was important there is we saw that translation from that early experience. We saw high muscle tissue concentrations, 42,000 nanograms per gram. Just to put that, what does that mean in construct? It was 20 to 30 times higher the level of exposure than what's seen with conjugates. So what it told us was, again, much like we saw in our preclinical experiments, this oligonucleotide, this compound was getting high levels of muscle concentrations. We saw that we were actually hitting, saturating in the skipping, that 53% skip transcript. So not only was the drug getting into the muscle, it was getting into where it needed to go in the muscle to skip that transcript that's required to generate protein. We saw high distribution, and this is really a unique characteristic of what we see with our oligonucleotide, not just to the muscle fibers themselves. And you hear a lot of the conversations about exposure to myofibers or muscle. But what was really unique about the distribution is we're getting into muscle stem cells. And we'll share a lot about that as we go forward across these studies and what that could ultimately mean in muscle repair, regeneration, and ultimately muscle maturation. Importantly as well, it was safe and well tolerated, and there were no SAEs in this early exploratory component of the study. So it met its core criteria. We didn't see protein at that stage. And as we had conversations, one of the expectations would be that dystrophin at 6 weeks would be unexpected. We then ran that into what we call part B of the study. And this was the opportunity to actually study this medicine over a longer period of time. And we'll talk about 2 time points in this portion of FORWARD-53. There was the interim, it's 24 weeks, so that's the 6-month time point. And what's very unique, and we'll talk about that in the context of FORWARD-53, is it's one of the first real longitudinal studies where we can look at, and we'll share some of these data later, where we could look at biopsies across time points to really get a sense of what's happening in muscle with the production of protein and the distribution over time, meaning being able to look at samples at 6 weeks, 6 months, and then ultimately 48 weeks, so nearly a year following treatment. So this was the interim data that we had shared. We shared this on a webinar when we had that data at the 24-week time point. And what we saw at that time point was one, consistency of dystrophin, so above 5% with a mean of 9% dystrophin protein. So again, we saw good concentration, stabilization. So actually dystrophin, the exon skipping stabilized, protein started to be produced. It was safe and well tolerated, no SAEs. But what we also saw, and there's some of these examples to the right of the screen, is we saw that we were getting into these muscle stem cells. And we shared these data where drug exposure was getting us not just into the myofiber, but into those stem cells. And what we could start to see now on muscle tissue was actually muscle fiber regeneration. So we'll talk a little bit shortly about what repair looks like in Duchenne muscular dystrophy, but the ability to start to shift to muscle fiber regeneration. Importantly, too, is these fibers were regenerating. We saw improvement in blood biomarkers like CK. And so the ability to see that these fibers, as they were being restored, were actually translating into measurements of muscle improvement. The other important thing in a longitudinal study like this is we could look at the tissue half-life. And what we had is a tissue half-life now of 61 days, which means that the chemistry in getting us a lot of drug in the right compartments also supported a profile of monthly dosing, meaning the exposure were there necessary to stabilize and sustain that. So the purpose as we think about the next phase of the study, this 48-week time point where we've seen protein, we've seen skipping, we've seen muscle concentration and distribution, was really thinking about the importance of what would we want to see after nearly a year on therapy. And it was very important to us with these early signals that we saw that we should be able to start seeing that transition to improvement in muscle health and functional outcomes. Those were going to be very 2 important drivers for us in continuing the program. So with that evaluation, that's exactly what we saw as we jump into the 48-week update. So very importantly, WVE-N531 remains safe and well tolerated with continuing no SAEs, so no serious adverse events. We see sustained consistent dystrophin expression averaging about 7.8% between the 24 and 48 weeks, with 88% of the boys above 5%. And we talk about this range because what we do see is stabilization of the dystrophin protein and recognizing, what consistency looks like between Western blots. And we'll talk about that as we see a very much stabilized dystrophin protein. We also see, and these are the important data with consistent dystrophin over this sustained period of time, is that we saw statistically significant reductions in muscle fibrosis. This is a remarkable finding in that fibrosis is usually an end event. And so being able to, and we'll share some of these data, see the reversal of fibrosis really is a big shift in what this could mean for muscle fibers. We also saw that those reductions weren't just in fibrosis, but also in CK levels. And this was driven by a decrease in inflammation, in cell death. And what we'll show you too on looking at the muscle biopsies is a transition from a regenerative state, meaning muscle fibers repairing and turning over, to one where we're actually starting to see mature muscle fibers, meaning the muscles actually repaired, restored, and looking more like mature muscle cells. We also saw that if we did all of these pieces, we would expect, as we said before, to start seeing some trends on clinical outcome measurements, particularly because of the small size of the study. What was remarkable is we have statistically significant and clinically meaningful improvements, so 3.8-second improvement in time to rise versus the natural history. And we also see improvements in other functional benefits and other measures, including the North Star. And we'll share more about those details. So as we think about the population on a study, it was important, as we heard from the community moving forward, that we increase the age range in the studies, and we'll continue to explore these in future studies. So the age range of the boys was between 5 and 11. The rest of the criteria were pretty consistent in terms of steroid utilization, and all boys on the study were on a stable steroid regimen. Continues to be safe and well tolerated at 48 weeks. Again, we had no SAEs. All events were mild to moderate. The one severe non-drug related event, just to highlight that since that's there, was a case of viral gastroenteritis that self-resolved and was believed to be second to norovirus. So again, nothing drug related and continues to have a very favorable safety profile. So as we move forward into dystrophin and the protein expression between the 24 and 48 weeks, we saw this range between 9% and 6.4. These are within the error ranges of the Western blot assay. The assay has a variability rate of about 30 to 35%. And in a challenging way, was never really designed well to move between studies and try to do comparative work. We actually ran to feel comfortable with that, and we shared this during the data release, orthogonal assays that allowed us to study all of these biopsies on the same gel to reduce that variability. And actually, there was a high correlation coefficient, meaning that they were related between the 24 and the 48 weeks. So that gave us actually more data points with which to realize that while there could be variability in dystrophin averaged 7.8%. And importantly, too, and as we've said a number of times, it really is important to look at the consistency of that. And I say that because as we think about designing future subsequent studies where you want to look at larger powering, it's important to see that the effect size of that dystrophin is distributed more equally across the subjects on the study so that patients on the study have the same levels of achieving that greater than Becker-like 5%. And that's important as we evaluated this study. So again, 88% of boys on study achieved greater than 5% dystrophin. But what does that mean? And I think sometimes we talk about the protein in a very singular basis, meaning what's the mean dystrophin level rather than how's it being produced, where is it being produced, and how can that ultimately change potential outcomes? And I think what's unique about where we see dystrophin being produced, and this is a highly differentiated feature of our designs and chemistry on our exon skipping medicines, is that we see this unique distribution beyond the myofiber, so the kind of the mature muscle cell, into these muscle regenerative stem cells. So if we think about what happens in healthy muscle, is there's a pool of regenerative cells that sits in the muscle. When that muscle becomes injured, the stem cell gets activated. It can form a new muscle fiber, and that muscle fiber joins the rest of the muscles in that fiber group and take on that structural effect. In the absence of dystrophin, you end up in a kind of a vicious cycle where these stem cells are activated, but they're unable to fully repair that muscle. So they get pushed into this reparative cycle. And that's where you can see that fat, scar tissue, and inability to actually build and sustain muscle. And so what's very unique is if we could actually restore dystrophin into those cells at that point of restoration, then that enables those cells to go down a mature pathway and shift. And that's exactly what we saw. So on the left are the biopsies from the boys, and these are all matched where we had biopsies from boys from the 6 months of the 24-week study and the 48-week study, and we could do comparative analyses of these two sets of biopsies. And what was really important to us was a couple features. One, there are these blue kind of purple dots, and those purple dots are the inflammatory infiltrate. And what we can see between the left and the right is that in the match samples, you see a decrease, and we'll walk through some of the quantification work in those inflammatory infiltrates. The second is we can see a decrease in necrotic cells, so cells that are undergoing cell death. And importantly, we can see more mature muscle structure, meaning the muscle fibers themselves are forming more functional structure. And that was unique to see, not just in one example, but across the study between the 6-month time point and the 1-year time point, that we could look at improvements in muscle fibers and muscle structure. Now, we also like to see how do you quantify that, because sometimes looking at cells become harder and you kind of step back and say, do you see this on a larger scale? And we did that. As we did our quantitative analysis, we could see the shift in both inflammation between the 6-month time point and the 48-week time point, and on cell death. So again, reinforcing what we were seeing visually on the cells in a quantitative way. Now, all of those improvements also would be important to see reductions in CK levels. And we saw a reduction in CK both against baseline. And importantly, as we looked at the natural history cohorts from the CPAT natural history cohort, we could see a statistically significant decline as well on CK levels. So importantly, we're starting to say dystrophin lining up to improvement in muscle fibers, reduced inflammation and cell death, but also translating to improvement in CK levels. Now, moving past those biomarkers and saying, well, what's this difference between a regenerative state to a mature muscle state is the ability to actually look at what happens in those muscle fibers over time. And what we could see was that, one, the density of these repair cells were decreasing. So what was happening now is there was less of a requirement. So they could go back into their normal state where they wait to be signaled and turned on. So that's an important state. And then secondly, and this is a very important measurement of maturing muscle fibers, is the shift from central nuclei, so these kind of nuclei that sit in the center of the cell, to moving to the periphery. And that's what happens when you get more mature, healthy muscle. And so again, we could see the core changes that happen within muscle from that regenerative, reparative state to that mature muscle state. So really important delineation. Now, that's all about muscle repair and getting better in terms of becoming more mature. We could also do a quantitative analysis where we could look at the fibrosis in the muscle. So this is the scarring that can happen in muscle that actually impedes its ability to function. And what we saw, which was remarkable across all the boys between 24 weeks and 48 weeks in the study, was that there was all a decline in that level of muscle fibrosis. Now, some of these changes could be starting as early as before the 6-month time point. And that what was so interesting was to see this shift between 6 months and 1 year, that this was, not an individual occurrence, but really did occur across the study. And so this is the first time anybody's been quantitatively able to show a reversal of muscle damage with fibrosis during a clinical treatment period. So moving from repairing muscle now to thinking about how does that ultimately translate to clinical benefit? And in this case, these were boys that we could continue to compare to the exon 53 cohort. So we had a very well-matched natural history cohort that we could use from a baseline characteristic standpoint to assess clinical benefit. And what we saw there was both a statistically significant as well as a clinically meaningful. So the MCID, or the minimum clinical important difference in this cohort, was 1.4 seconds. So we could see a substantial improvement in clinical benefit of boys treated during this 48-week time point. And this is a substantial magnitude, particularly when we look across other studies, and in particular at this 48-week early time point. So again, highly encouraging to see dystrophin translating the improvement in muscle fibers, translating to biomarkers, and then ultimately translating to clinical measurements. While not powered and usually takes longer, this is the measurement on North Star. So we saw a 1.2 point improvement. And we would expect this to continue to improve over time and with treatment. But again, highly encouraging that it continues to trend in the same direction as time to rise. We're also interested in measuring grip strength, and we'll continue to do this. And we did see improvements in grip strength during the study. And we do think, as we think about the compendium of clinical endpoints, it's just important for us to think about the totality of distribution of the medicine, where that's going, the muscle groups that can be improved there, and how to continue to find ways to measure improvements in a variety of muscle. Now, in addition to us seeing objective clinical data that we can measure, the feedback that we've received from our investigators was highly encouraging from boys on the study. So it's always nice when we get feedback of how the boys on the study are doing. It's nice when we can put that together with objective clinical measurements as well. And that convergence of what we're seeing in some of those measurements, like time to rise, are being conveyed across activities of daily living and things that I think all kids want to do, which is to play and to see that translate. So highly encouraging as we got that feedback. But as I said, it's nice to see this in the context of clinical data that we can measure as well. So as we think about continuing to move the program forward, it's important for us to look about what the profile is that we've delivered on. And as I said, we've delivered consistent dystrophin across boys, so 88% above 5%. We see the ability to get into both myofibers, but importantly, muscle stem cells, and that muscle stem cell delivery is very unique with the chemistry that we're using. Favorable dosing frequency, we are, and we'll talk about our plans as we go forward towards registration with moving to monthly dosing, given the remarkable tissue half-life we have. So we do believe this will be a monthly IV infusion. We continue to have a favorable safety profile with no SAEs. Again, we now see favorable evidence of functional benefit that's significant. We see reversal of disease progression in muscle, so being able to see that change in fibrosis and improvement in muscle tissue quality. And then as we shared in our preclinical data, and I think this is something unique as we continue to look forward in the study, both in continuing the work that we're doing in the ambulatory setting, but very importantly in the non-ambulatory setting, is the distribution that we have in heart and diaphragm that actually exceeds that preclinically what we see in skeletal muscle. So the opportunity to really think about broadly of where we can get dystrophin production as we think about designing future studies, continuing to look at respiratory and cardiac function. So as we continue to move forward, the chemistry for exon skipping that we're progressing does enable us to address the 40% of the DMD population with the exons that are highlighted, that are skippable, that we have, and being able to continue to explore others, and we'll talk about that work. Next steps for FORWARD-53 is all boys in the study are now consented in receiving monthly doses and have moved on to the extension portion of the study, so that continues. In addition, and to continue to support the monthly dosing regimen, we will be expanding the FORWARD-53 study to add a monthly continued cohort. So that'll be an additional cohort on FORWARD-53 that will be testing a monthly dosing regimen and continuing to generate those data. We'll also be submitting the NDA, so that's the new drug application for N531 in 2026 to support accelerated approval with monthly dosing. And we're actually plan to go back to the agency with these new clinical data. So at the time we had our discussions with the agency was with the 6-month data. So that was a dystrophin-driven conversation. With these new data and obviously with the clinical data in hand, we do plan to go back to the agency and continue those discussions on filing. Additionally, we're doing our work on those other exons to advance them, and we do expect to submit multiple clinical trial applications for our other exon skipping candidates in 2026. So a lot of activity for the WAVE team, but as I said early on, all of it's been inspired by the work that we've been doing with the community. And to that standpoint, I would like to thank all of the boys, families, clinicians, those involved in the clinical trial who make what we do possible and enable us to actually look forward.
Thanks, Paul. I think for now we can kind of open it up to the community for questions. I think a good place to start is if we go back to slide 5, we can maybe walk through some of these slides while some of the questions come in and some of the other ones we have submitted from before, just to make sure everyone is kind of understanding the pieces as we go through. So I think, yeah, it was back on just
All
to
right.
start with the preclinical. Yeah, no problem.
Yeah.
Just because again, some of these things, I think, some of our families have been in the space for a while, familiar with this. Other families might be newer, and some of this can be challenging. So even at the preclinical model, so kind of the significance here, talking about the double knockout. This is different from kind of the maybe the typical MDX mouse that families might have seen data on. We just talk about how those two are different.
Yes, no, and we've done a lot of work in the MDX mouse historically too. And so just a quick discussion because models, I would say models are important so long as how they're designed and what they teach you. And so the MDX model is commonly used. It has a knockout of one part of dystrophin and another knock-in of utrophin. So the model, because of that, the utrophin can support the survival so that in terms of doing work, those mice can survive a lot longer to be able to do work and studies on dystrophin production. I think what we've learned over time and some of the challenges with the MDX model is because there's that conflation of two different proteins that can both impact outcomes. Um, we were very interested in setting kind of what I would call a higher bar on the survival side, meaning these mice die, again, like I said, at 8 weeks, because in the absence of dystrophin, they undergo a rapidly progressive course, essentially an accelerated course of Duchenne muscular dystrophy within an 8 week time point. You'd imagine that rapid progression. And so therefore it amplifies meaning if you have an effect, then it's hard to see an effect there. But when you do, you can have confidence that it's coming from the underlying dystrophin protein because that's all that the animal is missing. So there's an importance of every model has its own strengths. There's always liabilities in moving from one species to another as you go from mice to non-human primates, ultimately to humans. And so our
Yeah.
view is the mouse data is purely about understanding how the medicine works, but it's one aspect to it.
No, that's helpful. I think it's good level setting for everyone. In this instance, there isn't a utrophin that can work as a surrogate kind of in place of the absence of dystrophin. So not just showing that you get expression, but that it can translate. And then we hope that this translates as we scale up to humans. And then just similarly for maybe families who aren't familiar with the non-human primate, that is not a Duchenne specific model. So these are not animals that have a form of Duchenne. It's going into healthy tissue. So that's why you're kind of just here looking at concentrations and not things like dystrophin production. And then I think on the next slide, so you kind of walked through some of the data that you've shown to date. And I think was here just for a point of clarification because you talked about this on this slide and then at the end, and just so kind of maybe families are aware, you talked a little bit about the half-life here, supporting that for N531, you can move to monthly dosing. But the data that you're showing here and then that was shown today was all while these patients were on kind of this every 2 week
That's
period.
right.
And again, kind of the data you're showing here to what you were saying, this is part of kind of telling the story. We want to get to functional data, and that's really important, but these are kind of those early markers to show that what you're delivering may be having an effect.
Exactly.
And then I think if we go forward to where you were able to show some of that data, I think it was maybe slide 13. Yeah, so here. So I think one thing, so you showing kind of the dystrophin expression by Western, and it's muscle content adjusted. And I think that's a term that we've been seeing more in releases from companies. And so just want to make sure, kind of families have some understanding of what that is compared to maybe non-adjusted when you're looking at these.
Yeah, I think it's interesting. I mean, as we know, the company that's been leading in the commercial space with DMD is really, pioneered the thought process around this. And I think we always approach things, I call it with a healthy degree of skepticism of how should we be evaluating it. But this is one where actually, after a lot of analyses, makes a lot of sense in looking at muscle because when we think about how you measure dystrophin, looking at the muscle content because so much of what you take around different biopsies and different samples have different levels of fat, scar tissue, and other substrate versus muscle fiber. And so the idea around thinking about muscle content adjustment is trying to create a normalization factor that while you look at different samples, does adjust for the fact of the muscle content within that particular sample and analyzing dystrophin to create some way of normalizing across various samples how to look and compare at it. And so as we go forward, like others, I think it's an appropriate way to think about characterizing dystrophin
Yeah.
and
And I think it's also
standardize.
too, right, because you're going and you're taking these biopsies from these boys, and you're not doing multiple biopsies and multiple muscles all throughout the body, right? So you're kind of taking this one biopsy. That's kind of the shot you have to do the analysis. And so as we'll see in some of those slides later, that we know that there's fibrosis and fat infiltration. So you're trying to look directly at just what is the muscle tissue? How much are we getting to those pieces?
That's right.
And then on the slide on the right-hand side, so looking at kind of the variability of expression noted, I guess two things to hear. So there's kind of this line set at 5% and kind of looking at 5% is a marker. So can you talk through just kind of where that number comes from?
Yeah, there've been actually some really interesting work in publications looking at various degrees of Becker muscular dystrophy at different levels of dystrophin production. And what was interesting is there was a remarkable change between 0 and 5%. And then there was a small change between greater than 5%. And so there's a lot of desire, I know, because when we're quantitative, people think, well, more is necessarily better. But people try to follow that number. But the data suggests that once you're around that 5% threshold, even above 0, as we've seen with other exons, but above 5, that you're now in line. I think when we think about that line, and the reason why we use that not as a singular point, but looking at it in the collective, is when we do talk as a community about the Becker phenotype and how that's different and we have to remember that that 5% number is really spread through all the cells in the body. So it's not just a 5% within the cells where you get an exon skipping therapy, and it's really looking at holistically. And as we spoke about later, a while ago, that concept that even in those patients, that 5% is in the regenerative cells. And so if we have to think about the totality of it. And so it is important less about the amplitude. And, there's a whole range of where you get exposures, as you pointed out. But for us, it was really about do the majority. And that was what we set as the bar for the last portion, the 6-month study that we did. Do the majority of boys experience that greater than 5%? Therefore, you can feel like the drug in all of it, because you're right. When we're talking about biopsies, because they are precious, and we do realize that you get very few and you want to limit the amount of muscle that gets taken during any clinical trial, you do get one very, very small window to actually look at what that potential medicine is doing. And so what's encouraging is when you see it consistently across boys in a study means that you're actually getting that exposure in a more uniform way where it can actually be having the effect.
Oh, great, thanks. I think that's really helpful context for families as we kind of talk through these. And then just from another standpoint of we can see this and this is going to be with any drug that we look at where there's going to be variability in kind of patient response. So, and I know this is a very small N, these are early studies. Have you had a chance to kind of look across the different participants to try and interrogate, whether other age, the different, there's a lot of mutations that are amenable to exon 53 skipping, any of the
Yeah.
factors that might contribute to the kind of the differences that we see in individual patients.
No, and we did that because to your point, one of the things we look at is are there different mutations or deletions that we even within 53 that and it was remarkably consistent. I think what we all know a bit is that there is inherent variability too, both in you have to remember the muscle that you get from each of these studies creates an inherent variability that makes it harder to compare across. And then you've got the assay itself that has some inherent variability in it. So it's why I speak to consistency more than an absolute. And really why we were really interested in seeing the translation to clinical, where eventually we move past some of the variability just on a dystrophin number by itself and really ask the question of what is it, what is this protein doing? Are we actually creating a functional protein that's restoring muscle? But there is inherent variability every time you take a sample and what that's going to look like between patients.
And then if we move forward, I think again this is helpful even just on the explanation because I think some of this, if we go back one to the satellite
Yeah,
to the stem cell piece, just helpful context because I think this is something that we started to talk about this a little bit at conference last year, and there were I think some differences in understanding because some of the families that maybe have been in the space longer have heard different things about why the stem cell issue and challenges with Duchenne, and so. This is based on some more recent kind of work done over the past few years, looking at the importance of having dystrophin in those stem cells and performing a function. And so this is something that we necessarily haven't seen with a lot of kind of the exon skipping therapies to date, but have been something that we've been kind of looking for. And so this gets, I think, to what you were describing a little bit before in terms of not just getting to the muscle, but the importance of getting into that for enabling some of the regeneration.
exactly.
Um, Here, so I'm just gonna raise this in case any families had any questions. So, week 24, so that is, patients have been on drug for a little while. Do you have baseline biopsies that you can at some point compare from that to week 24 to week 48 as we kind of look at as dystrophin has been being produced longer in patients?
Unfortunately, the study didn't have baseline biopsies
Okay.
for each one because with the concept of two, and that's what I think made this study unique in some ways. But as you point out, the challenges, which is we actually had the opportunity to look between 6 months and a year on
Mhm.
therapy, but with following the and we understand two biopsies kind of is the maximum for
Yeah.
patients. We have to think about where we do that. What we did see, and we shared this back with the 6 month data readout, is we do have biopsies across patients from 6 weeks to 6 months. Those who are on the study, and you could see that early shift. That was highly encouraging back last year with the 6 month data when we compared the 6 week biopsy, which is more or less an early baseline to the 6 months on treatment, there was a difference. So when we start to line that up, we can start seeing that shift from, as we were saying, you kind of get to that regenerative state at that 6 month period to a much more mature state when you get to the 48-week period.
Great. And I think these type of slides I think are really helpful for families just to help visualize because it's I know on the next slide there's some of the numbers and the shifting and
Right.
it can be kind of hard in the abstract to look at that and go, all right, so they're saying it's less severe, but how do I actually kind of see this? And I think these are really clear representations of the kind of changes that we're seeing, which again we hope then translate to the functional benefit. And so on the next slide, I think it was you were just kind of walking through some of these. So, um, can we make, I don't know that maybe families have seen slides like this kind of from this minimal to mild kind of scoring and going across. So we can maybe just walk through this for the inflammation and cell death.
It's much more common in other diseases, like thinking about liver disease and inflammation, or people look at shifts like in NASH, non-alcoholic steatosis, ways of thinking about how do you look at changes in inflammation. So if we're on the left column and you go top to bottom, you start looking at what would be severe and kind of the fours, the threes, and where does that shift? And so this is, I always call it's semi-quantitative. The histology for me is always the much more helpful way to look at kind of muscle in general. But it lets us have, and pathologists have a scoring system when they look at, in this case, inflammation and inflammatory cells. You look at those markers, those kind of purplish cells, and they rate them in a very standardized field. And you go, okay, this one was kind of severe or kind of worse inflammation. And now you score the new samples. And so you can do more direct comparisons across patients. And the same thing in terms of cell death. Here they're looking at what are called necrotic cells or cells that are going through that apoptosis and dying. And so you can count up those cells in a field and say, okay, what do they look like within these various fields? So again, ways of kind of putting a more numerical assignment around what we just saw on the prior slide. But at least that continue to say that what we're seeing and what we believe we're seeing actually now can relate to a quantitative measurement.
Great. Okay. That's very helpful. Thank you. And then at the next slide, so this is kind of walking through the CK, which is again good, helpful as a nice biomarker for us, and I think one that the community is very familiar with. So just here, the comparison is to this DMD natural history cohort. Um, Can you talk about why you had to use a natural history cohort for this study?
Yeah,
For
so on
the
the left is
comparison?
baseline, and without a placebo, we needed a natural history cohort to follow. So what was important is we think about those inflammatory markers and we look at CKs. We know that all the boys who we were measuring were on a stable level of steroids, so they weren't changing their doses or other medicines while they were getting treated. So we saw against baseline the decrease, but then to your point, we had to compare against the natural history cohort to get a comparison of what would be more of a placebo cohort.
And then I think for families as well who maybe aren't familiar, so CPATH, that's the critical path. And they have a number of data sets, both natural history studies, placebo arms from clinical trials that have been contributed from academic centers and companies over time to help kind of give us a good understanding. So it's not just necessarily a single study that it's compared, it's some pooled data from kind of those groups. And then I think maybe slide 19. Just walking through, yeah. Yes, so I think this was just, again, the visual is, I think, very helpful for families. So seeing kind of the fibrosis here between the week 24 and the week 48. So similar to what we saw before with those other, the other slides you were showing. So at that 6 week time point and then continued on until week 48. Can you just, I think, talk a little bit about the importance here of the ability to where we can maybe have some of this reversal of fibrosis in a way that we can't with some of the kind of the fatty tissue that is kind of the next, we think about as the next stage as we talk about kind of fibrosis and then its eventual replacement.
Yeah, I mean it's wonderful we line these up because to your point on, the discussion or the evolving discussion on the importance of regenerative cells and muscle and fixing muscle cells and improving muscle health. One of the other features you want to see now is over time with a decrease in inflammation, cells not dying, which kind of stops that cycle, is do you see this shift in fibrosis back? Meaning now do you get more healthy fibers that are now uniform in their organization? So, as we start talking about clinical activity and what's going to improve clinical function, it's having at the heart functional muscle groups that can do the things that they need to do. And so by moving away that fibrotic component, and that's what's so unique about being able to look at this quantitative metric. So in red, if we look about the difference between week 24 and 48, just that's how the scoring happens to give the data points that go in those middle graphs that actually you can plot is a way of digitally capturing fibrosis. So looking at those stains in red and looking at the decrease in that fibrotic tissue. And so this method of now seeing more uniform organized muscle is ultimately, as you said, one is what translates actually to improve muscle unit function. And that, should be what translates ultimately to the clinical changes that one could see.
Great, thank you. And then I think on slide 21, when we start to show some of the functional data, um, again, just pointing out, so this is a little bit different. So again, a natural history cohort because this is not a placebo-controlled study. And so, but this natural history cohort is different than the one that was used for the CK cohort.
Correct. This was from an exon 53 clinical trial placebo group, so it follows a natural history cohort that actually is very, very well matched to the patients on the study.
And then I think just important for families as they're looking at that in both the time to rise and with the NSAA is there is this there's we can see kind of the separation here between these groups. Still is some you know change in terms of the time. So, the patients still are losing a little bit of time in that, but at a much slower rate, which, this is something we've talked about at advocacy conferences before with the community is the importance of kind of stability and slowing disease progression, and that these are really important markers. And always important and glad to hear that you'll be able to kind of re-engage conversations with the FDA, but we always want to make sure they understand as well the significance of this to our community, that these are kind of really important markers for us. And then I think on slide 25, you had kind of a highlight of a number of the things that the summary slide essentially of a lot of
Yeah.
this data, which was, encouraging. I think one of the questions that I, had is, and I know this is often the question with this is, does it, and you showed in the non-human primate data, so some evidence that you think that it's getting to the diaphragm, that it's getting to the heart. are you either with this open label or in future studies going to kind of look and try to monitor that and test and see if there's improvements in either respiratory function or cardiac function?
Yes, I mean, it's in this study. I mean,
Okay.
at this study, and fortunately, boys weren't yet at a stage where we would expect that progression to come on, although we were measuring and showed that it wasn't detrimental, but not at a stage yet where we'd expect to see separation. But because we do have that from baseline now at a year and continuing to follow it, and given the extension studies continuing, we will look to continue to measure lung function as well as cardiac function. And then as we work on subsequent studies, there's been a lot of discussion at WAVE of thinking about the opportunity we have to explore again, both the ambulatory and non-ambulatory setting, ways to really distinguish and delineate a lot of the impressive preclinical data we have around the distribution, to run those in clinical studies.
Great. And then on the next slide, and I think some of the questions we had coming in around potential studies. Yeah. So there was just a question around kind of with this, expanding FORWARD-53 to include additional boys on this monthly dosing. Just question on kind of, what the sites would be for that. And I think, families who are maybe interested in participating, what countries that'll be in.
Yeah, and I'll separate the FORWARD-53 with the expansion versus next steps for the study and opportunities going forward. So currently, FORWARD-53 was conducted outside the United States. So the initial expansion will obviously be within the existing sites outside the United States that were involved in that study. Our intent in following our conversation with the FDA following the 6-month data is about the work that we will do globally. So that's inclusive of the US. And so that will be built around continuing to build for both the confirmatory study design that we would start anyway before that submission. And so that work's underway, and we'll be definitely engaging with sites and the community as we get that study up and running. And that will be a study on monthly dosing. So again, very similar to the expansion cohort that we'll be running that longer out. So we do have plans for a global study, US and ex-US, and we'll be staying close with everyone as we design that study.
Okay. And then I guess whether this is in FORWARD-53 or for the future, there was a question that came in around, given that there are other products for those who are maybe amenable to exon 53, if someone was on that, is there an ability to participate if they have some kind of washout of their current treatment?
The protocol did allow for that. We didn't have in FORWARD-53, despite the protocol allowing for it, we didn't, and that was because outside the United States, there's far less people on exon 53. But the protocol was built to allow that. So if we were able to expand FORWARD-53 and get that up and running before it completes in the US as well, that would be an inclusion criteria that you could be on a previous exon 53 skipper and be washed off.
Great, thank you. And then another question along those lines would be, it was a question of, could a patient be on an exon skipping therapy if they've received gene therapy? And I think there's kind of two different ways. There's the, is this possible? And then there is the, clinical study design space for kind of WAVE.
Yeah, I think to the first point, the answer is absolutely yes. I mean, I think they're mechanistically distinct, and there's every possibility that these two medicines could work in concert and not disrupt one another. Different is the clinical trial design, and one of the challenges on clinical trial designs is answering the question of, is that benefit? And now having seen clinical improvement at, again, the 48-week time point in this much smaller study, being able to do that again as part of a larger study, you want to assure that the production of functional dystrophin, where you're seeing this effect, that you can prove that's related to the study drug. And the challenge in that setting is that there is the confounding effect of the gene therapy and the exon skipper to being able to establish that relationship and signal in the context of a clinical trial. And so that doesn't mean that there aren't opportunities for clinical studies that look in combination of gene therapy and exon skippers and how they could potentially work together. But in a clinical trial specifically as it relates to functional improvement driven based on exon skipping, it's very difficult to design a study that can distinguish if you're on a gene therapy, whether that's having a confounding effect. So we're having those conversations. We know there's a lot of desire, particularly in some circumstances where we're hearing from the community about the potential of waning efficacy. And can you add the exon skipping therapy on? I mean, I think we understand that these are all very real questions in understanding biology, and may very well warrant clinical studies to be able to explore that. But we have to make sure we look at each study that it's going to answer a very specific question, recognizing that we have to go into a regulatory environment that needs a very specific answer.
Right. Which I think, the importance there, and we talk about often, is, kind of the challenge with the clinical design and making it something that is rigorous enough to kind of get us to an approval so we can then get the therapy to more patients.
That's right.
It's a balance that's, tough to strike sometimes. And kind of along those lines, we had a question around, would this be a product that could be suitable for someone who's a manifesting carrier? And again, I think this is the two-part of, there is the, could this work for someone who is a manifesting carrier who's amenable to exon 53 skipping, and then would there be opportunities in a WAVE clinical trial?
Yeah, and I'll separate the answer on those two and it's not dissimilar from I think the prior answer, which is potential, yes. If there's any person amenable to exon 53 skipping to generate a skip transcript to make a protein would be amenable for these therapies, 53 in this case, but other exons as we move forward. Again, within the clinical trial context, as you pointed out, you try to get some homogeneity on the clinical trial so you can answer a question in a very specific way that ultimately is very well said, and we agree very much, is all about how do we get access. And getting access means getting to that approval state where we can assure that it's reimbursed so patients can get it. And so to move through that regulatory paradigm, we need to have as consistent a clinical trial as possible to generate those data. But it doesn't mean we can't look at other studies that would be separate from that study to be able to explore those opportunities. And we think a lot about that in kind of two contexts. One is obviously being able to look at, as you pointed out, and in the heart and diaphragm, and are there studies that we can do that are really designed to see those impacts and that effect, which we believe is different. The other piece that's unique is getting into that regenerative cell, those stem cells very early on, has us thinking a lot about getting earlier in the treatment setting. And with all the work that's been done by the community on newborn screening and kind of getting to the heart of understanding where you can intervene earlier and being able to intervene very early on those regenerative cells to kind of restore dystrophin into muscle fibers a lot earlier. Those are two areas that are of high interest to us, and we just have to think again about how do you run those studies to deliver those experiments in the setting still of moving the program forward to assure it gets to patients.
And then we had a question come in because I think this is probably a little bit maybe challenging sometimes to understand because I'm gonna ask a little bit about the chemistry. So this is like how can we tackle this? So there was a question coming in asking about, we've seen some recent presentations from other companies who are starting to use conjugates to PMOs, and we've seen it both with peptide conjugates, antibodies, antibody fragments. So a question of, is that something that you're thinking about? But I think there's also, what you showed today was evidence that you're getting good exon skipping, you're getting into the cells, you're having good expression, and we're seeing some functional benefits. So can we just walk through again how kind of the chemistry here for why you're able to kind of take this approach without having to necessarily maybe go the conjugate route?
Yeah, it's a wonderful question. I know highly confusing as we see a lot of different types of conjugates, a lot of different what it's the conjugate stuck to, and we talk about PMOs, and we talk about phosphorothioates. Wave's backbone is what we call this mixed backbone. It's unique to when we started the company, well over a decade ago. And at our heart, we're an oligonucleotide chemistry company. And so if we think about the heart of making a medicine, at the heart, they're all chemicals. You put together a design and you think about how do you design it uniquely. And at Wave's core chemistry was this ability to really create what we call stereopure, chirally controlled. And this means that when we make an oligonucleotide, which is very different than how others build them, we get one single medicine. So we actually have a chemistry that in the field, and I'm not going to make it more complicated on a chemistry level, but when you put together the building blocks of an oligonucleotide, if you look at a standard oligonucleotide that has, let's say, 20 linkages, because it's easy for math, you get about 524,288 different chemical substrates that are all in that. When you get a wave oligonucleotide, when we design a single oligonucleotide, you get one. And so the advantage of that we've seen over time in history within the space is when you have a mixture of medicines that you're delivering, they can all have different unique pharmacology associated with them. You don't see it because everything's studied in an average, but they can have different potencies, different safety profiles, different stabilities. And in general, you talk about it as if it's one, but it's really a mixed average. And you're delivering that with either a conjugate or by itself. With Wave, we could define the medicine as one single entity, which means we could change the backbone in different ways. We have unique chemistries on the backbone. In this case, what's called PN. We use that across a variety of our formats in the central nervous system, so in the brain. We've used it in the liver, and we use it now in the muscle. And what we've seen with this is it gets us, as Eric pointed out, really high drug concentrations in the muscle. 30 times more than what's seen with the drug conjugates. And so it told us that we didn't need to add a conjugate to it. Now, the beauty of having a single drug means the same drug that we test in vitro is the same drug we can study in animals like non-human primates for distribution. And then we could take that same drug and use that in our clinical trial and understand how that works. Now, where the advantage of this really comes is now when we think about moving to another exon, the ability to say, okay, we understand what this backbone did, this one drug, and how it worked. How can we take that same chemistry that we put on the back of that same exon and just apply that to another? And so what it lets us do is be able to take that shared learning from one program to the other to be able to continue to build. But it without complicating more, it really does get that back to the heart of our unique chemistry capability, where we don't have to take on the liabilities that come with another chemical, right? An antibody or a Fab or a peptide that can have its own associated liability. So it really lets us focus on the single medicine.
That's really helpful. Thanks, Paul. I mean, it's challenging, I think, sometimes because
It
we
is.
do all these different ways, and everyone kind of has their different approach. But that's helpful. And I think the last question here was there was a question around kind of age for the trial, both kind of I think asking, if there's going to be any changes to the current age cohort for the expansion of Forward 53, and then I think what I'm going to throw in as part of this too is as we're getting newborn screening kind of in the United States, and we have a number of states that are doing this, we like to think that earlier intervention is going to provide, greater benefit for patients. And so is there any interest or, plans for kind of maybe looking at some younger age cohorts than, are kind of typical for 5, typical age of diagnosis range?
So Forward 53, we have that range. There's been some discussion about for the expansion. So to date, though, we have that established range, and yeah, I think that's consistent. I think very much to your point, as we think about the next design, we are thinking about, whether or not there are opportunities, particularly moving into the earlier setting, given the data that we've seen. And so that's the work that we'll be doing, and obviously we'll be interacting with a number of you in the community as we plan that study. But we are taking that into account. And again, whether it's part of the next study, which would be a potential confirmatory study, or whether or not that's part of a separate study looking at the earlier setting or later, we'll be evaluating that.
Oh, great. Thank you. I think that's all the questions that we had in the chat, and we're just about at time. So I just wanted to say, thank you for joining us and walking through this presentation and taking the time to kind of go through some of that in great detail to help make sure everyone who's on and watches understands the data that was presented. It's really helpful. And then just as a reminder for everyone who's on, we should have this up online within the next week. So if you need to go back and re-listen to any parts, or if you have friends or family who would benefit from seeing this, you can direct them to PPMD's website. But just thank Paul and Wave for joining us today.
Thank you, Eric, and most importantly, thank you all for joining. I know you've got a lot you can be doing, and we're happy to spend the time. And we're always open to hearing your thoughts directly too. So we appreciate this format to interact, but please reach out, and we're always happy to engage.
Thanks, everyone.
Thank you.